The process of copying DNA, this is known as DNA replication, occurs in the interface of the cell cycle. DNA replication is a very complex structure of DNA, which is anti-parallel double helical structure. Deoxyribonuclease nucleic acid, when replicate, it helps with the DNA helicase enzymes. So I want to explain you here the structure of the DNA, which is simply when we enter the helicase enzyme, it means there is a breakdown of DNA molecule into single strand. When it breaks down the DNA helicase in the presence of DNA helicase enzymes, there is a breakdown of single strand, and another enzyme, due to this enzyme's nucleotide formation, known as DNA polymerase. DNA polymerase from the new nucleotide into the 5 to 3 direction. What is 5 to 3 direction? Here, the structure of DNA, when it replicates, the direction goes this side. This is called leading strand. Direction which is 5 to 3. And when the nucleotide continuously direction with the 5 to 3, within the 5 to 3, this is known as lagging strand. In the lagging strand, the formation of Okazaki fragment. Okazaki fragment, when combined with each other, when alternate with each other, it's due to the ligase. Here, the structure of DNA, when Okazaki fragment, one Okazaki fragment, two Okazaki fragment alternate with each other, this is known as ligase. Okazaki fragment. In Okazaki fragment, as we know that there is a complete structure of nitrogenous base, which is adenine, guanine, thymine, cytosine. 4 bases: adenine, guanine, thymine, and cytosine. After this, another process, when due to one single strand form a new DNA, this is known as semi-conservative. What is semi-conservative? When new nucleotide forms with the help of one single strand. This is one single strand. Here, sometimes DNA structure damage. When DNA structure damage, there is abnormality, there is dysfunction. Here, the nitrogenous sugar phosphate, it damage. Cancer happen due to this structure damage. Sometimes in the internal side of DNA, which we said it's a nitrogenous base, okay? And adenine. So the connection between these two bases is not normal, it's abnormal. So how can we repair the DNA structure. When we encode the gene into the DNA structure, the structure will be normal after this step. Structure will be normal. Guys, I hope you enjoy this concept. Thank you so much.